Okay, you did this activity in class in small groups. However, I want to make sure that when you're looking at these traits, at these letters, that everybody remembers that we're talking about genes, different versions of a gene found in a chromosome. So when you're talking about having a big Y and a little y, those are the two possible genes that can segregate into one of the four gametes that any individual makes after it goes through meiosis. And when you're talking about having two genes that segregate together, or they segregate again independently as far as which one you get when and where, you're talking about having another gene, another locus, that has, again, either a big R or a little r, different versions of the letter, different versions of the genetic code. So this gene in this particular chromosome codes for something. It's called R, in this case round or wrinkled, wrinkled being this version of the gene here, all right, and round having this version of the gene here, both coding for shape. This is the gene for shape. One version, because it's ATGTT, gives you a wrinkled outer coat, and one, it's ATTGT, gives you a round coat. Whatever the sequence is, there's a small difference in the code that gives you a different, that codes for changes the protein that changes the shape or function of whatever it is that it's coding for. In the case of Y, we're talking about a yellow and a color, and one version of the gene being yellow, the other green. And again, each of them sorts independently depending on which of the gametes it ends up in after meiosis. So dihybrid crosses reveal the law of independent assortment, the way I explained to you in class. And remember what we talked about in meiosis, when we went over meiosis, is this idea that the genes sort independently of one another. That it doesn't matter, they're not going to sort together the way they do in mitosis. The whole purpose of meiosis is to make sure that things get scrambled up with crossing over, with independent assortment. These things increase the ability for an individual to increase variation, genetic variation, and the way it does that, you're able to mix up the genes that you have. You're not creating any new ones, you're just mixing up the ones you have. Although mutation does happen, the key is by just the process of meiosis, you're able to do this kind of independent assortment, all right? So Mendel was able to figure that out without knowing anything about genes, because he called them particles or factors, and he knew that they segregated independently based on the math, on the results of his experiments. Now, when we went over the dihybrid crosses over winter break, there was some question as to what was happening between the parental generation and the F1 generation. And remember that in the parental generation, you were dealing with purebred red-green and purebred wrinkled yellow, and that gave you this heterozygote round yellow. Now notice that the wrinkled and yellow were purebreds for each of the genes, and so was the round and green. But when we came down, when they were mixed together, they produced 100% heterozygotes. And if we did the Punnett Square, and you'll do it, you have already done it in the activity, well, you could do the Punnett Square. These are not dihybrids. These are purebreds, right? Or they're homozygous for the two genes. You'll notice that when you cross this, when you take this F1 and you cross one of these with another one of these, so that's what this demonstrates here, you end up with this ratio, 9:3 to 3:1. You always have a 9:3 to 3:1 when you have two dihybrids being crossed with one another. Remember that. I want to point something out here. You'll notice that there's actually 556 seeds. When we say 9:3 to 3:1, we're talking about reducing 315, 106, 101, and 32, reducing these numbers down to a ratio of 9 to 3 to 3 to 1. So if we take 32 and divide each of these by 32, we get 1339, and a total of 16 when you add these up. So these are the actual results, and this is the reduced number. So these remember, ratio should be reduced to its smallest form. Here is again the summary of Mendel's dihybrid crosses. I'm not going to read them to you. You've already read them in class, I hope, and you've taken notes on this. Here again are the ratios produced per trait, the shape, the color, and again the demonstration of 9 to 3 to 3 to 1. Once you've reduced the numbers 315, 101, or 108, 101, and 32. And notice that they're not exactly 9 to 3 to 3 to 1. They're about 9 to 3 to 3 to 1. If you did the math, you'll see round and wrinkled. It turns out 423 to 133, so that's 3.2 to 1, and this is 3 to 1. 3.2, I think you'd agree, is pretty close, especially when you're dealing with these giant numbers. You'd expect there to be... You can't expect that these crosses to come out perfectly every time. The idea that you get about a 3 to 1 ratio every time is impressive. This shows you the Punnett Square that you would have to make in order to look at this dihybrid cross. And you know that when you mix these two dihybrids, big R, a little y, little r, big Y, and you come up with you take these are not dihybrids, these are homozygous, big R, little y, etc. for this. That this is the only gamete that they can make. There's only one possible gamete they can make. This plant could only give you a little r, and this plant can only give you a big Y. No matter what happens, it's all they can give. And this plant can only give you a big R, and this plant can only give you a little y. That's all it can give. So what you must produce 100% is this heterozygote. You take these two heterozygote, you cross them with heterozygote or dihybrid, and you cross them with one another, you get these 4 possibilities. Where did I get these? From FOIL. I'm not going to do it again, but that's what it comes out to. And here, again, it would be FOIL. Now you'll see you get 1/4 of these, 1/4 of these. Now they put a semicolon in between them. We usually just write big R, big Y, but the semicolon's fine either way. It's just a notation. And you'll see that these are the possible phenotypes, right? You can get round yellow, round green, wrinkled yellow, wrinkled green, and these are the ratios you end up with 9 to 3 to 3 to 1 of each of these phenotypes. Assuming you're crossing, this is what you'd expect to get, assuming you crossed this dihybrid. Let's take a look at the answer. As you see, the answer is exactly that. If you counted up all the round yellows, you see 1, 2, 3, 4, 5, 6, 7, 8, 9. If you count up the round green, 1, 2, 3, you count up the wrinkled yellow, you do 1, 2, 3, you count up the wrinkled green, you'd have 1, so 9 to 3 to 3 to 1. All right, so there again, another breakdown of phenotypes, and here it shows you the genotypes. These are the phenotypes that are produced. These were the numbers, these were the ratios, 9 to 3 to 3 to 1. And that's really the key here, all right? So if you're looking at dominant, how many are dominant to green, you'd have to look at a 3 to 1 ratio, and that's the others 3 to 1 as well. So the two heterozygotes are 3 to 1, right? The homozygotes are 9 for dominant, or not, yes, whatever you get. Anytime you have a dominant in either one of the genes, you have about 9 of them, where if you have a homozygote recessive, you only have 1 of them. There are 3 hints or 3 rules of thumb that you should be looking at before you try to do a dihybrid cross or be thinking about when you're doing one. The first thing you need to do, and you always remember, is you're looking for all combinations of gametes that are possible. And remembering that you only get one allele per per gene in a gamete. So where you in a parent you'll have two, a big A, little a. In a gamete you're only going to have one. You're either going to have the big A or the little a, the big A or the little a. You'll have the big P. You know what? Let me go ahead and use the letters that they're using here. You're either going to have a big P and a big T, or you're going to have in one gamete on a chromosome, or you're going to have a big T and a little P, or you're going to have a little T and a big P, or you're going to have a little T and a little P. And which one of these you're going to have, and how did I figure these out? And again, you go ahead and do FOIL. So those are the four possibilities you could have if you had two heterozygotes, or they were heterozygotes for both genes, or both genes. The parents were heterozygotes for both genes. But when we look at this, that's not the case with this particular problem. And this is what they're telling you. This is one of the things that they want to make sure you understand. When you're doing a dihybrid cross, you don't necessarily have to do a full 4x4 16 squared dihybrid cross. If you look here, this is homozygous for the T, and this is homozygous for the P, homozygous dominant for the T, homozygous recessive for the P. So when you're making the total possible gametes, and you go ahead and do FOIL, what you find out is that there's only two possible gametes that this cross could give you, right? This being crossed with this could only give you one possible outcome, all right? Meaning that this parent could only give you big T and big P, right? Big T and big P. This big T with this big P. And it can then a big T with a little p. That's this one. So this parent could only give you that. Where this parent here could only give you big T with little p and little t with little p. So this parent could only give you this. These are the only two gametes that this parent can give, because that's all it has. Now, when it does meiosis, of course, there's two of these and two of these, and two of these and two of these, right? Because you have 4 cells. But in all 4 cells here, depending on which of the 2 crosses we're talking about, in the 4 cells, there's only going to be 4 possibilities. This parent can only give you big T, big P, or big T, little p. When it does the crossing, when it does meiosis, it'll give you 4 gametes that look like this. But these are the same. These two are the same, and these two are the same. And since we're going to reduce the numbers anyways, it doesn't make sense to go ahead and do the crosses with 4x4. In other words, instead of doing this kind of thing, and doing it across this in this direction as well, you would rather probably, and I hope you would say you would rather do a cross where you're just doing half the work. So when you end up looking at the zygotes, you have only 4 possible zygotes that can be made from a cross with 2 where you have a homozygous for one gene, for the T gene here, and homozygous for the P gene here. Because you only have 2 possible gametes here, and there's only 2 possible gametes for the other parent. So you have 4 possible zygotes once you did the cross. If you did it here, this is what you end up here is you end up with four different ones, right? Over here you have 16, but once you reduced it, there'd be a lot of doubles. In fact, it would be exactly double. So you'd end up with only having 4 different ones anyways. So it really doesn't make sense to do this 4 by 4, which is what they're saying here. Not all squares are necessary when you have a homozygous on one of the genes. And I think you've seen that in some of the sample problems we've gone through. In fact, one of the other videos I sent you was discussing what you do when you have a homozygous parent. One of the students asked, why don't you do FOIL with the homozygous parent? And the answer was there was no need to, because there was only one possible gamete that it could make. In this case, there's only 2 possible gametes these two can make. So you need clearly to state the phenotype and genotypic ratios. And so let's take a look. So this is a dihybrid cross. This would be the dihybrid cross that you would have gotten from these 2 parents that I just did the FOIL method on. And you'd have this going on, right? And here you have, and over here, and here. All right. So you'd have these 4 possible gametes to work with, and of course you'd list what they are, tall or whatever the particular phenotypes are. They're defined by the problem. There you have it. And here's the genotypes for each of the results. You have one of these, one of these, one of these, one of these, and you have tall and purple and tall and white. This is the way is one way you can represent what you see. And of course, these numbers need to be reduced. Here, you don't need to reduce them because you left out the other 2 repeats, but you'd have to reduce them if there are repeats. See, here's the genotypes. Okay, so Mendel's second law, the law is the law of independent assortment, and I've said it many times. There's no other way to say it. It's just a random assortment of possibilities, which is why we use the Punnett Square. And here is an example of what we're talking about when we're talking about independent assortment. Each of your genes can go into 2 places. Will go into 2 places, and which gamete it ends up with? Does this big Y always go with big R? No. It could randomly go with any one of these when it creates its gametes. What is this process that you're looking at? You're looking at meiosis, right? That's what we talked about. That's why you were supposed to learn it. That's why we went through it. So when you're looking at one nucleus turning into one cell turning into 4 cells, each of them haploid, in other words, 2 copies going to 1 copy of each gene, then in which gamete does any particular gene go, or any allele go? It's all random. Here again are your 4 gametes and the law of independent assortment. Now, when we're talking about a test cross, if you have, and what exactly is a test cross? A test cross is when you have an individual that's yellow and smooth, and you want to know, is this homozygous or is this heterozygous, right? You need to cross it with something that is homozygous or pure breeding, and see what you get. If this were big R, big Y, in other words, if this particular individual here was big Y, right? Big R, big R. If this was, it would still be yellow and round. It would still be yellow and round. But when I crossed it with this little y, little r, right, the homozygous recessive, then my products, these what I would get in the end would be all round and yellow, right? Because every one of the zygotes would have a big Y and a big R, because that's all this parent would be able to give. But that is not the case, right? That's not what we have here. As it turns out, when I did this cross, and remember, when you look at a round and yellow P, there's no label that says big Y, little y, big R, little r. By doing this test cross, using a pure breeding wrinkled green pea plant, and I cross it with this round and yellow pea plant, and I get all these different combinations, that tells me that this individual is heterozygous. If it wasn't heterozygous, if it was homozygous, then I wouldn't get any yellow wrinkled, and I wouldn't get any green wrinkled, because these are recessive traits, right? They have to have the little r, little y in order for there to be a double little y and a double little r. All right, we'll talk more about test crosses later, but it's key that you understand that idea. And this also links into the pedigree discussions that we've had. As we do these crosses and look at patterns and segregation, what you notice is that when you're dealing with one gene, you tend to, and you're dealing with phenotype, you end up seeing a lot of 3-to-1 ratios. And when you see that, when you have an F2 generation, that's two homozygotes being two homozygous parents being crossed, and you cross F2 generation, what you end up with is you end up saying, when you have big A, little a, cross with big A, little a. That's what they're talking about there. When you have that, what happens is you end up with only one of these, 1 out of 4 here, 1 out of 4 for the homozygous dominant, and then 2 out of 4. There's 2 of these. But if you look at the phenotype, what you see is 3 phenotypes that are all the same. Why? Because remember that the heterozygote is the same phenotype as a homozygote when you're talking about dominant and recessive traits. This kind of inheritance, if you have a big A, you are big A. And if that happens to be, yellow, then they're all 3 are yellow. If that happens to be green, they're all 3 are green. If then it happens to be tall, then all 3 are tall. Whatever it is, if you have the dominant allele, you have that phenotype. So 3 of the 4 have that particular phenotype. And only 1, the homozygous recessive, is going to have the phenotype that is recessive. So when you're talking about phenotypic ratios when dealing with one gene and a dihybrid, and you have a hybrid, for the parents are both hybrids, you end up with a phenotype that's often 3-to-1. You often end up with 3 of the dominant showing up and only 1 of the recessive. One of the recessives. Now, when you're dealing with genotypic ratio, you tend to have 1 to 2 to 1. So 1 big A to 2 big A little a's to 1 little a. So you tend to have a 1 to 2 to 1 ratio when you're dealing with one gene, 2 hybrids, they're crossing. This is the pattern you see over and over and over again. And the phenotypic in a test cross, and I want to remind you what a test cross is. If you have this thing that's, let me go ahead and use green. If you have a green, a green smooth pea, and you want to know, is this thing a purebred for green and smooth? Or is it a hybrid for this particular color and shape? Well, in order for it to be green and smooth, it would probably be little y, if it were homozygous. Well, that would have to, you know what, instead of saying green, let's assume that in this case, yellow, little y equals yellow. It's not true usually. And big Y, anything gives you green. Okay. And I'm just going to stick with one trait here, one gene. We could do it with two genes. But let's go ahead and, since we're talking about test crosses here, we'll do it with one, and then we'll go ahead and do it with the other. So with one gene, if we're looking at this creature that has the seed that turns out to be green, so this seed could be big Y or anything. How do we know? Can we tell what this second gene is? Can we tell its genotype? Not just by looking at it, but we can take that seed and we can cross it, that zygote, and we can cross it with, if we cross this Y blank with something that's definitely a true breeding plant. So let's say we cross it with a yellow. All right, a yellow. What would we expect? Well, if we have big Y blank and we have little y, we would expect that for sure half of them are going to be green, right? Because you're always going to have that big Y. But then the other half, we're not sure. If this variable, this second unknown, this blue star, were big Y, then you have 100% green peas, right? Because if both of these were big Y's here, if both of these were big Y's here and here were big Y's, because this is a big Y, then all four of these would be big Y, little y, which would mean that all four of them would be green. If, however, this blue unknown were a little y, then you'd have half of them green and the other half yellow. Does that make sense? So if you're crossing a yellow or a homozygous, our test cross is taking some unknown and crossing it with a homozygous dominant or homozygous recessive, and then seeing what kinds of zygotes you get. And if all your zygotes are one type, then you're probably dealing with this particular unknown this genotype is homozygous for that gene. If you get 50-50, then you know that individual cross is not. It's heterozygous for that gene. All right. So you either get 1-to-1, 1-to-0, or 1-to-1, and this is 1-to-1. If this was a heterozygous, you'd get 1-to-1. You'd get 2 yellows to 2 greens. That's 2-to-2, but reduce it, it's 1-to-1. If this ended up being a big Y, so it's homozygous dominant, then you'd have 100% green and 0% or 4 green to 0 yellow, and so you'd have a 1-to-0. Okay? All right, let's go ahead and delete that and look at a double or a dihybrid. So in a dihybrid cross, if we had the same kind of question, except now we want smooth or wrinkled, does this particular green pea have the gene for wrinkled? And let's say wrinkled it's smooth. We know it's smooth. We can see it's smooth. But does it have the gene for wrinkled? So again, let's say little r is wrinkled and big R, little r is smooth. So if we cross this green P with this, so this is green, so we know it's green, it's Y, and then we don't know what the other one is, and we know it's smooth, so it's definitely a big R, and it has, again, an unknown. I shouldn't have put the cross there. That's so silly. If that's the genotype that we don't know that second one, and we cross this with a true breeding yellow smooth P. Okay, let me go ahead and draw that in. We cross it with a yellow smooth P. What would we expect to see? Well, what are the possible gametes this one can make? This one can only make one gamete, little y, little r. So that's easy. That makes our life a lot easier. And the next question is, how many different gametes could this one make? Well, it would depend on what was the second gene. So if we saw, let's assume then that this is big Y, little y, big R, little r, then we would see 4 different possibilities of big Y, big R, and we'd see big Y, little r, and we'd see little y, big R, and we'd see little y, little r. So those would be our 4 possible gametes. And so what would we expect to see? What would we expect to see if we cross this, if this is heterozygous and we cross it with this homozygous recessive, we would expect to see 100%, 100% green smooth. Oh no, we wouldn't expect that. Sorry. If this was heterozygous, we'd expect to see green, 1/4 green smooth, 1/4 green wrinkled, 1/4 yellow smooth, and 1/4 yellow wrinkled. So we would expect to see 1/4 each. And of course, if we know this is y, little r, so that doesn't change. But if there's an if the y were homozygous and the r were heterozygous, then it could only give big Y. So there would only be 2 options, right? It would be big Y, big R, or big Y, little r. So you'd only get 2 possible zygotes, big Y and big R, little r. So we'd end up with a green green smooth, and over here we'd end up with a green rough or wrinkled, right? So you'd have 50%, 1/2 green smooth, right? And then we'd end up with 1/2 green wrinkled. 1/2, in other words, I just want to make sure this is clear. This would be 1/2 green smooth, and it'd be 1/2 green wrinkled. So we'd end up with a 50% chance here at these positions, where if it were, and that's if you did a big Y, big R, unknown, right? Big Y, little r, and you cross that with a little y, little y, little r, little r. The test cross again is going with the recessive. You cross it with a recessive to see what happens. So that's two possibilities. This is one here, and this is two. There's a third possibility, right? At least. So that was homozygous. Over here, it was we had a heterozygote crossed with our recessives. Here we had heterozygous for one, homozygous for the other. I'm not going to do 3 because 3 would be the same thing, except that it would be this would be the homozygous, and then this would be the recessive. I mean the homozygous. So you'd make this big Y, little y, and this big R, right? And then that would give you your homozygous, or little r. And you can go ahead and do that cross, and you end up with 50-50 either way. So this is when this is heterozygous and this is homozygous, and if we made this big R, we know it's not little r, right? Because it's green and smooth. So if it was little r, this would be wrinkled. So we know it's not little r. The question is it big R, little r, or big R? So if we made this big R, and this big Y, little y, and crossed it again, we'd end up with 50-50, except they'd both be wrinkled, and one would be green and one yellow, right? So that's another possibility. And then, of course, the final possibility is the fourth possibility. And that's if, in the fourth possibility, you have both of them being homozygous dominant, and that's crossed with the heterozygous, the homozygous recessive. This is what's called a Testcross. That's the definition of a Testcross. This one can only give one possible gamete, and this one can only give one possible gamete. So then we would have big Y, big R. This is the only gamete it could give. Crossed with big Y, big R. Crossed with little y, little r. Only one box it could be, big Y, little y, big R, little r. So they're going to be 100% green and smooth. So by doing this cross and looking at what are the phenotypic ratios you get, you know the genotypic ratio of the parent. Because if the parent, you know what the phenotype is, it's green and smooth. It could be two possibilities. It could be big Y, little y, or big R, little r. It would still be green and smooth. It's actually 4 possibilities, right? Three possibilities. It could be big Y, big R, big R. It could be big Y, little y, big R. It could be big R, big Y, or it could be big R, big Y, or big Y, little y. So 4 possibilities, 1, 2, 3, and 4. The first one we looked at was what if they're both heterozygous, so big Y, big R, little r. And you would have 4. You'd have 4 different things that come out. You'd have 1/4 of your seeds would be this, and 1/4 would be this, and 1/4 this. That is a possibility, and if it is, then you know that this is heterozygous. If it comes out anything like that, then you know it's heterozygous. If you have, then you try to do a cross, and you don't. If you had big Y, big R, little r, then you get 50-50. 50% green smooth and 50% green wrinkled. If this were big R, big Y, little y, and you test crossed it, it'd be 50-50 again, except it's be the wrinkled would be, they both be wrinkled because this is big R, or they'd both be smooth because this would be big R. So it would be green, it would be yellow, green, smooth, and yellow, smooth. So in any case, if you have a 50-50 ratio, you know that you're dealing with one of the two genes being homozygous and the other heterozygous. If you have 100% come out in a certain way, in this case green, smooth, then you know you're dealing with a homozygous dominant for both genes. These are all three of Mendel's laws clearly stated for you. You've gotten a copy of this hard copy, and you now have the digital copy as well. Now, when we're dealing with probability, we have to think a little deeper than just 1 out of 6, for instance, in a die. What are the chances of me getting 1 out of 6 or 2 out of 3 or what have you? We have to try to apply this to Mendelian genetics. All right, so when we're looking at expected probability, what you're asking about or what you're thinking about at this point is this idea of how many times do we expect this to happen and how many opportunities are there? So Punnett Square kind of looks at that, and it does that by defining the total possibilities as 4. All right. And then how many times do you expect something to happen? In this case, how many trials is 4, and what are the probabilities that how many times do you expect that to happen? That's what gets filled in here. So what are you dealing with when you're dealing with a gamete? You can either have a big A or a little a, or a big A and little a. A big A and a little a, in which case this would be dominant, this would be heterozygous, this would be heterozygous, and this would be homozygous recessive. And so given 4 opportunities, 1, 2, 3, 4, what is the likelihood of getting 1 big A, 1 out of 4, right? The total is 4. So we would represent it as 1 out of 4. Or you could write it as 25%. Take 1 and divide it by 4. It gives you 0.25 multiplied times 100. So let me be clear. Take that, divide 1 by 4, and that'll get you 0.25. Take that and multiply it times 100%. And that's going to equal 25%. Think of this as a unit. All right, so the total possible, all 4 out of 4 is 100%, or 1. 4 divided by 4 is 1. Or you could represent it as 100% by multiplying times that 100. That's what this piece here is. So let's take a look at an example here. You have 60 red gumballs, and there's 40 green gumballs. If you buy 1 gumball, what's the probability that it's a red one? Well, there's 60 gumballs, right? 60 red gumballs. There's a total of 100 gumballs, because 40 plus 60 plus 40 is equal to 100. So you have a 60% chance, or 0.6. When you do the division, you have 0.6. And 0.6 times 100% is 60%. Now, I did this on the board with you. I think it was the Friday, at least one class. I think I did it with 2 of the classes. I did it on the board with you, assuming that you had already understood this when we went through small groups. You should have paid attention to this and be able to understand that when you're looking at percentages and what are the chances or probability of something happening, this is what it's meant by. You take the total number, the number of chances of events, the predicted number that you could happen, and how many total possibilities there are. So 60 out of 100, and that's your probability of getting a gumball any given time. Now, when you're talking about the product rule, you're thinking about two independent events. Independent, wherever you heard that word before, even in this particular video, I hope you remember the law of independent assortment. So this is why we have to think about independent events. The probability of two independent events occurring together, you multiply them. And so we call that the product rule. So you take, what's the probability of A happening and the probability of B happening, and you do that in percentages, left in decimal form. You take those two decimals, you multiply them together, and that gives you the probability of both of them happening together. So if I roll two dice, what's the chance of getting 2 5s on these two dice, as you see here? So that would be a 5 on one and a 5 on the other. You see the word and. So if you're looking at the word and, then you multiply them together. Well, how many sides are there on a die? There's 6 sides, so the total would be 6. And how many 5s are on there? There's only 1, so it's 1/6 chance. And there's 2 die. What are the chances of both of them happening, one and the other one happening again? I'd have to take 1 over 6 and multiply it times 1 over 6, which I hope everyone knows is 1 over 36. And that's my odds. Those are my odds of getting both of them happening together. And if you want the percentages, you take this 1, you divide it by 36, and you multiply by 100. 1 divided by 36 is 0.0277777, repeating. So round it up to 0.028 times 100%. That would be moving the decimal twice. So it'd be 2.8%. So 2.8% that this would happen the way it is, to getting 2 5s in a row. And here's the math of it. There's 1/6 chance of getting a 5 on one die. There's 1/6 chance of getting a 5 on the other die. You multiply those two together because you're looking at and. What are the chances of these two happening together? You get 1 out of 36. And if you take 1 divided by 36 and multiply it times 100, you get about 2.8%. That is your product rule. In the sum rule, you're looking at or. What are the chances of getting this or this to happen? So let's take a look at what this example. I think it's best to look at an example. If you roll 2 dice, what are the chances of getting 2 5s or 2 6s? Right? And a 5 on first die and a 5 on the second die, or 6 on the first die and a 6 on the second die. All right, so what are the chances of having that happen? Well, we already said it'd be 1 in 36 chances of getting 2 5s in a row. Well, it'd be the same chances for these two to happen together, right? One and the other. So the probability of getting 2 5s is 1 out of 36. And the probability of getting 2 6s is 1 out of 36. But now all I have to do is add them together, right? So if I add them together, I get 2 out of 36, or 1/18, because I can reduce that. Remember, we're always looking for the reduced version. But I can always divide the top by the bottom. And if you take 2 out of 36 and divide it by 1 over 18, you're still going to get the same number. Or you say 2 divided by 36 and you get a decimal. You take 1 over 18, you get a decimal. You're going to get the same decimal. All right, you're probably going to get 0.056. 0.056. Something. It'll probably continue. So if that happens, what you end up with is that the chances of all this one or the other happening is 5.6% chance. So moving on, sum rule in the multiplication rule, sum rule, and the product rule. What is the probability of getting one green and one red ball if we have 2 quarters? This can happen in 2 ways. First, you can get a red first, then a green first, then red first, or a red and then a green first. So there's 2 ways to get that, right? There's the only 2 ways to get it, having 2 quarters. When not specifying the order, we must figure out each way of getting the outcome. So if somebody doesn't give you an order to figure out that probability, you just say, well, it could happen green over red, green first and red first, and red first and green first. So moving on, we have 60. Again, we still have the 60-40. The probability of getting green and then red, you use the product rule. What are the chances? Is 0.4 because it's 40 green and there's 60 red. That is gives you 24% chance. What about red first and green first, then green? You have a 24% chance. Again, you're using the product rule. So the probability of getting one red and one green gumball and one green gumball, right, is the probability of it happening in this way, the probability of happening this way added together. So 0.24 plus 0.24, or 48% chance of getting a red and green. So when we look at the Punnett squares, in a way, when we look at a Punnett square, it's a way of depicting the product rule. All right. So Mendel's rules are the rules of segregation, which means they're independent, right? And so they're equally likely to occur. There's no one is linked with any other. So for a cross, what we do is we just say, well, what are the possibilities of getting a big R? Then there's little r, there's big R, little r. And we say, well, this times this is 1/4, this times this is 1/4. We don't do the multiplication, we just move the letters around. But it turns out that what you're doing is you're using the product rule, and using this Punnett square is helping you reduce the amount of calculations you have to do. But understand where it comes from. It comes from this idea that when 2 things are equally likely to happen, and you want to know what are the chances of these 2 things happening together, having a big R and a big R, then if this is half of your possibilities, this half, in other words, there are 2 big R or little r, and you can either give a big R or a little r. So what are the chances? 1 of getting a big R, 1 out of 2. And that's the same for this big R. So multiply them together, 1 out of 2, 1 out of 2 is 1 out of 4, or a quarter, or 25%. And then, of course, you see that ratio of 1 to 2 to 1 when you're dealing with a single gene. What are the chances of getting 2 heads in a row with a fair coin? In other words, a regular coin, head or tail. 2 heads in a row. So getting a head, and then getting a head again. Well, it's 1/2, 1 out of 2 here, right? How many times how many possibilities do you expect to happen? 1. Out of how many trials? 2. Right? Same here. Total possibilities 1 out of 2. So 50-50. So those 2 happening together is 0.5, which is half, right? Times 0.5, which is, hopefully is 0.25. So you end up with the number 3 being the correct answer. What are the chances of rolling a 1 or 2 with a die? 1 or 2. 1 or 2. Well, to get a 1, it'd be 1 out of 6 here, right? To get a 2, it's only 1, 2 on that die, so it's 1 out of 6 there. So what are the chances of getting 1 or 2? 1 out of 6 plus 1 out of 6 equals 2 out of 6, reduced to 1/3. And they didn't reduce it here. They kept it at 2/6. Now, on a test, they might reduce, on a standardized exam, they might reduce it to 1/3. If the parents of a family already have 2 boys, what's the probability that the next 2 offsprings will be girls? Well, for the next 2 offsprings to happen, that's 2 trials, right? So what are the chances of it being a girl and a girl, right? So you already have, what are the chances of all this happening together? We have a girl and a girl. It's one half times 1/2, because it's the next 2 being girls. 1/2 times 1/2 equals 1/4, or 1/4 of your chances. They kind of tricked you on this a little bit when they said already have 2 boys. This does not impact this, right? So unless they ask you, what are the chances of having, I'm not sure how they would phrase it in any other way, but there's really not a good way there's no way no really good way of explaining this, except to say that if they say if you already have 2 boys, the 2 boys, they're not really asking you what are the chances of getting boy, girl. They're asking you the chances of getting the next 2 times this happens for it to be 2 girls in a row. This having 2 boys does not influence the 2 girls at all. So the question, the way it's phrased, is the answer would be 1/4. If they ask you what are the chances of having 2 boys, then 2 girls, then you'd have to do a little more of the math. It'd be a little more complicated. So here, consider 2 genes with 2 alleles. You have a big Y and a little y, a big R and a little r. So cross 2 purebreeding lines. So here's a purebred line, here's a purebred line, and you get your heterozygotes. All the F1s are going to be 100% heterozygous, right? Because this can only give a big Y. This can only give a little y. This can only give a big R. This can only give a little r. So you take your heterozygotes, all the possible gametes. There's 4 of them, all right. And when we do our math, what are the chances? So we do our math. What are the chances of getting a big R, big Y gamete? That's 1 out of 4. So 1/4. 1 gamete out of 4 are going to be big R, big Y. So here you see the 1/4 is all the way across. 1 out of 4 here, 1 out of 4 here. That there's 2 heterozygotes being crossed together, 2 F1s being crossed together. Here is your classic heterozygous chart that you're always going to see over and over again, and you're always going to get the 9, 1, 2, 3, 4, 5, 6, 7, 8, 9 yellow rounds, again phenotype only, 3/16, or we could reduce, I can't really reduce 3/16, 3 out of 16 round greens, so 1, 2, 3, 3 out of 16 yellow wrinkled yellow, and 1 out of 16th green wrinkled. And so you have that 9-3 to 3-1 ratio that you'd expect in a dihybrid cross. All right. Okay, again, this is just looking at looking at the product rule. Just think of it as a mathematical problem. You can see that 9-3 to 3-1 is expected, right? Because if you have a 3/4 of your gametes are going to be big R, big Y, right? Big R, big Y. 3/4, and you take 3/4 and you multiply them times each other, and you get 9/16. So you'd expect that it'd be 9 out of 16. So what I mean by that, and let's take a look at these 2 small Punnett squares. If you took this R, you independently made it a single hybrid cross. You see R, big R, big R. What are the chances of having big R blank? 1, 2, 3. As we said before, 3 out of 4, right? What are the chances of having big Y blank? Big Y blank, 3 out of 4. So what are the chances of getting big R blank, big Y blank, and big R blank and big Y blank? What are the chances of that? We would use our product rule. So 3/4 times 3/4, you'd expect to get 9/16. And indeed, you see that when we do the big chart. Well, what about little r, big Y, little y? Well, little r is only 1 out of 4. The other big Y blank is 3 out of 4. So what would you expect? 1/4 times 3/4 equals 3/16. This gene combination, 1 out of little y is only 1 out of 4. The other is 3 out of 4, 3/16 again. And what are the chances of getting little r, little y? That's 1 out of 4 times 1 out of 4, 1/16. So this 9 to 3 to 3 to 1 ratio is mathematically predicted using that product rule. Oh, here it is. Here's the answer to that. All right, so the next question is, what is the probability of obtaining a round green seed from a dihybrid cross? Now let's think about it. Round green seed. So green is little y, little y. It's the only way to get it. Round, it could be 3 ways, so it's 3/4. So to get round and green, you get 3/4 times 1/4 equals 3/16. Why is this blank? Because there's 3 ways to get round. We're talking about phenotypes. It could be heterozygote or homozygote. But when you're dealing with the little y, there's only one way to get green. A fraction of the progeny from the following cross will have a large, smooth, purple fruit. Large, smooth, purple fruit. What fraction? So large is big L. Purple is big P. Smooth is big S. So what fraction of this dihybrid cross, and there's 3 of them, look at it, there's 3 of them. What fraction of these are going to be large, purple, and smooth? Well, let's think about it. For it to be large, there's 3/4 of the possible Punnett Square here is large. Smooth, you can have, well, because, let me look. There's the trick. I was going to say 3 out of 4 again, but that's not right. The trick is that this is homozygous recessive. And because it's homozygous recessive, that means that you get only half of them. Half of them can be smooth, and that's these 2. Normally it would have been 1/4, right? And here purple, it looks like they're it's homozygous dominant for purple. That means they're all purple, 100%. So that's a 1, right? So there's your trick. You have a homozygous here for purple. That means all the progeny, 100% of them are going to be purple. Where this is heterozygous in both of them for large. So large, you have 3/4 of a chance to get large. You have 1/2 a chance to get smooth, and 100% chance of getting purple. So what are the chances of getting all 3 and one and? When you hear the word and, you're multiplying them because you want to know what are the chances of having all of them together. So 1/2 a chance for smooth, 100% chance for purple, and 3/4 of a chance for large gives you the total of 3/8. So it's a 3/8 chance and notice that this is a trihybrid. So these are 3 genes. The laws of probability for multiple genes. What's the probability of obtaining the genotype RR, YY, TT, SS? Now we're dealing with 4 genes. RR and RR, you get 1/4 this, 2/4 that, 2/4 here, and you do a Punnett square for each of those, okay, separate Punnett square. What's the probability of obtaining an individual with RR and YY and TT and little s, little s? The probability of events occurring together. There's the word and. So you just take each and multiply and times each other, and you get 8 out of 256, or 1/32, 1 out of 32 possibilities. And now we're not talking about 1 gene or 2 genes or even 3 genes. We are talking about 4 genes. And these 4 genes, the chances of getting heterozygotes for 3 of those genes and homozygote recessive for the 4th is 1 out of 32. And you can express it as fractions if you'd like. So what are the probabilities of obtaining a completely homozygous genotype? Probability of either events, one or the other occurring, right? So you have these. This is being crossed with this, which gives you these two. And it looks like everything's heterozygous. And what's the probability of obtaining a completely homozygous genotype? Probability of either or events occurring. Genotype could be, in other words, it could be this or this, because it says homozygous. There's 2 possibilities, homozygous dominant or homozygous recessive. So probability of getting homozygous recessive here is 1/4, 1/4, 1/4, 1/4, and homozygous dominant is 1/4 each. So what do we have to do? And it goes back to just the product rule and the sum rule. All right. So if you want to know what are the probabilities of getting the homozygous dominant, and then you figure out what are the probability of getting homozygous recessive, and then you take the probability of getting the homozygous dominant and add it to the probability of the homozygous recessive. So what are the odds that I'm going to get big R, and big Y, and big T, and big S? Well, there I'm multiplying. It's the product rule, right? What are the probabilities of me getting bigger, little r, little y, little t, little s? Now here again, I'm multiplying because I'm using the word and. But then I have to wonder, what's the probability of me getting this or this, right? One completely homozygous genotype. Well, then I'd have the probability of this plus the probability of that equals 256. 2 out of 256. That's you can multiply or divide 2 by 256, get the decimal, multiply times 100, and that's your percentage. All right, if we're taking a look at this cross, we're looking at heterozygote for each trait, and there's 4 traits. 1, 2, 3, 4. 4 genes, rather. 1, 2, 3, 4. So what is the probability of obtaining this genotype? So we're talking about a homozygous R and a homozygous S. Now, if we remember our ratios and how Punnett squares work, this gets to be a little easier than not. So I would suggest just writing out the simple Punnett square for just one of the genes. Because as it turns out, the ratios are going to be the same since they're all heterozygote, you see. You don't have to know as long as you know the ratios, it doesn't matter whether the letters are S, R, Y, or T, the ratios are going to be the same. So here you have big R, little r, big R, little r, and then little r. So you'll notice that the ratios are the same, the ones that we had discussed before. We have a 3:1 phenotypic ratio. We have a 1:2:1 genotypic ratio. So that this becomes handy. I like to draw it out once just to see it, but it becomes handy to know that you have a 1:2:1 ratio every time you have 2 heterozygotes. So it helps you when you're trying to do your mental math as you're jumping through the problem. If we cross this with this, what's the probability of obtaining a genotype that looks like this? Well, we have to remember we have to find out what's the probability of getting each individual one the way it is. So what's the probability of getting the R, big R? Well, that would be 1/4, right? What about big Y, little y? Well, there's 2/4, right? This would be 2/4 because if you look, but wait a minute, I didn't put Y up here. It doesn't matter, right? As long as you know that the heterozygous is going to be 2 out of 4 or 1/2, right? 2 out of 4 is the same as 1/2, but we're going to keep it 2 out of 4. And in fact, let me shorten this. Oh, really? Let me draw this a little differently. Just say all these the same. This is 1 out of 4. this here is 2 out of 4. Now, should I reduce it to 1/2? I could, but it turns out it's easier if I just leave it like it is because I'll be doing some multiplication soon. And here I have 2, again, it's another heterozygous, so that's 2 out of 4. And another homozygote, so I know that that's 1 out of 4. So what are the chances of me getting big R, and big Y, little y, and big T, little t, and big S, big S? Well, I would multiply each of these together. So 1 times 2 times 2 times 1, that's 4. And 4 times 4 times 4 times 4 is 256. So now if we reduce that, divide each of those by 4, 256 divided by 4 is 64. So reducing this, we get 64, 1 out of 64. So if we have this test cross, what's the probability of getting each of this particular genotype? I would take 1/4, 2/4, 2/4, and 1/4. That gives me 1 out of 64, and that is this answer here. Now, if we're going to do this cross, let's look at what we have before we look at what we need to get. So you see that they're each heterozygous for each of the 1, 2, 3, 4 genes, or all 4 alleles are heterozygous. All 4 genes are heterozygous. And here, what's the probability of obtaining one homozygote dominant, one homozygous recessive, and the other 2 heterozygous? Well, the easy way to do this, or easy way, we would go ahead and do a test, not a test cross, excuse me. We do kind of a little Punnett Square to remind us what kind of expectations we would have for each of the heterozygote crosses. What are the odds of getting any one particular one? And I hope you see that when we look at it, again, our phenotypic ratio of 3 to 1 is there. In other words, you get 3, whatever big R phenotype is, and 1 little r phenotype. Or you have the genotypic ratio of 1R, 1 to 2 to 1, right? 1 to 2 to 1. So you know there's 1/4 chance of getting big R, 1/4 chance of getting, or half a chance, 50% of getting big R, little r, and 1/4 chance of getting little r. So here in this problem, they're giving you the question, they're giving you this word or. So here you should know that if you see the word or, you should be thinking, right, adding. So here we're using the sum rule, right? But before we need something to add before we could use the sum rule, right? We can't add anything. We don't have any numbers yet. So how do we get the numbers? Well, to get the numbers, we would use our multiplication, right? Or our product rule. What are the odds for each of the individual genes? Well, to get big R, you'd expect that there would be 1/4 chance to get that. Each of the heterozygotes you'd expect to be 1 or 2/4 or 1/2. And the homozygote recessive is 1/4. As it turns out, this ratio ends up being the same. How's that possible? Well, the homozygote is always 1/4, whether it be recessive or dominant. It's only 1/4 chance. So this is 1/4. This is 2/4. This is a 2/4 chance. And this is, again, a 1/4 chance. So how do we find what are the chances of this happening and this happening and this happening and this happening so that we can end up with this whole thing, that genotype? Well, we take each of these and multiply it times each other. And so what we would end up with is at the bottom 256, and at the top we'd end up with 4 out of 256. And if we reduce it, it'd be 1 equals 1 out of 64, just like the previous problem. And the same thing would happen here once you get it all done. It's 1 out of 64. So it's a 1 out of 64 chance that you're going to end up with this particular genotype. 1 out of 64 chance you get up with that genotype. But what are the chances of getting this one or this one? So you add them. So 1 out of 64 plus 1 out of 64. And that's where I get my numbers, and that's how I get my solution. So that's 2 out of 64. Well, I can reduce that. 2 divided by 2 is 1, and 64 divided by 2 is 32. And indeed, there's our answer. 1 out of 32. So you see, it's all about using the sum rule and the product rule to get our answers. The key to these word problems is to try to figure out, to try to remember, where's the or being used, when are you adding, when are you using the sum rule, and when are you using the product rule. When you're using the product rule to figure out the ands, what are the chances of having all these happening together? I'm multiplying that, and I'm reducing, and I'm getting an answer. Probability can be used to predict progeny, but remember, we're talking about a large sample size. In order for the larger your sample size, the more accurate your probability prediction. Any given toss of a coin can be heads. You can get 10 heads in a row, but the odds of getting 30,000 heads in a row in a completely random flip, very hard, very, very difficult to perceive that happening. So the Punnett Square is a graphical representation of these possible outcomes. The whole point of showing you this is to show you the math behind the Punnett Square. The phenotypes are the result of the genotype in the organism. All right, so remember that more than one genotype can give you the same phenotype. So it doesn't matter if it's heterozygous or homozygous. And distinct segregation patterns result from monohybrid, dihybrid, and test crosses. So you get specific patterns in that 9 to 3 to 3 to 1 ratio. You get that 3 to 2 to 1 ratio. So you need to really, or 3 to 1 ratio for the phenotype in a single in a monohybrid cross. The 1 to 2 to 1 ratio when you're talking about the genotypic and genotypic ratio in a monohybrid cross. You have these patterns that you can learn from, and you can actually use them to more easily predict or answer questions as they come up. So that concludes our probability discussion. I hope that helped. If you have any questions of going over exactly what is the product rule, how we apply it, what's the sum rule, how to use it, you should be you're allowed to use calculators, so it shouldn't be too much trouble for you to use a calculator to answer a question on that. All right, well, good luck to you.